Hello, my name is Andrew Ribeiro. I'm the head of the Laboratory of Biosystem Dynamics of Tampere University, here in the Faculty of Medicine and Health Technology. In this work, I try to present the results of our investigation about the ability of the bacteria Escherichia coli to tune the distributions, the population distributions of RNA and protein numbers by using the dynamics of the rate-limiting steps during transcription initiation. All cells, including bacteria, constantly need to make decisions. They need to decide where they're going to, they need to decide what proteins are they going to produce, they need to decide what mechanisms are they going to activate or deactivate, etc. To achieve this, they have evolved gene expression regulatory mechanisms. So these are the mechanisms that allow them to tune how many proteins the cell has. From the point of view of the population, this implies that they're able to tune the distributions, the single-cell distributions of protein numbers. This can be done by, for example, tuning the mean rate of production of the proteins. In some cases, it's also possible to switch between opposite behaviors, from producing to not producing. It is also even possible to tune that this cohort of proteins produces, while this other cohort of proteins does not produce. For example, when they go from stationary to exponential growth phase, or vice versa. One example circuit that is capable of exerting two opposite functionalities is a genetic switch. Basically, it consists of two genes that mutually repress one another, which allows either one be on and the other off, or vice versa. In this circuit, the frequency with which the switch between the two states occurs can be tuned by altering, for example, the noise in the production of the proteins of gene 1 and gene 2. So far, as in the case of the toggle switch that we described, it has been generally assumed that noise in gene expression products is symmetric. That is, if a protein is produced too much, in another cell, there's protein that is produced too little, more or less by the same amount. In that sense, the noise is symmetric, which is either to one side of the mean expression level or to the other side of the mean expression level. Now here, what we investigated is whether it is possible to regulate gene expression in order to introduce asymmetries in the distributions of RNA and protein numbers of cell populations, and whether these asymmetries can influence the propensity, that is probability, of crossing a threshold in protein numbers. So far, as in the case of the example toggle switch shown, it has been generally assumed that noise in gene expression products is symmetric. That is, that the degree of noise below and above the mean expression protein numbers are identical. In this work, we investigated whether one can regulate gene expression so as to produce asymmetries in RNA and protein numbers, and whether these asymmetries could influence the propensities for crossing thresholds in protein numbers. These asymmetries are usually studied, for example, in economics, whether, for example, in a population, the money is distributed symmetrically. There are so much poor people there as there are rich people. How is the money distributed? Now, in E. coli, transcription initiation is a very complex process that has essentially two main consecutive rate-limiting steps. The first step is the formation of the closed complex between the RNA polymerase and the promoter region of a gene. The time that this step takes is determined by the concentration of RNA polymerases and by the affinity of the RNA polymerase to bind to the promoter. This can be regulated by, for example, changing the promoter sequence, by the presence of activator molecules or repressor molecules, which increase or decrease the time that this process takes. The second step is the open complex formation, where the DNA has to be unwound open for reading. It is after this step that once the RNA polymerase escapes from this region, that transcription elongation can occur. Now, the time length of this step can also be regulated and is also sequence dependent. Meanwhile, the other steps in transcription are much faster than this, and thus, in general, in normal conditions, they do not control significantly the time interval between consecutive RNA production events. Interestingly, based on the model that one generally assumes, one can expect that by tuning the relative duration of the two steps, as I exemplified here in this work, one can significantly affect not only the mean production rate, but also the shape of the distribution of time intervals between RNA production events. We investigated if and how this can be achieved, and we present here a brief summary of our results. For this, we mostly made use of microscopy measurements of single RNA production events in live cells. We use fluorescent tags that allow visualizing when a new RNA is produced, as exemplified in the movie, and each new spot is then visible, and it represents a single RNA, which is detected by the MS2GFP tagging technology. We also used flow cytometry to verify that the proteins that are produced from those RNAs are consistent in numbers with the measurements of the time intervals between our production events. So if we have long intervals, we expect very few proteins to be produced. If we have short intervals, we expect far many proteins. Finally, we made use of several other methods for validation of the results, which we will be describing throughout this work. From the measurements of time intervals between RNA production events, we extracted two things. First, from measurements in many cells, we obtained the distributions of time intervals between consecutive RNA production events from many cells. From each such single-cell distribution, we calculated the mean, the coefficient of variation, the skewness, and the kurtosis of that distribution, which are known to be able to measure the asymmetry of the distribution. We also used the method to extract the average time length of the events before commitment to open complex formation and after commitment to such open complex formation. That is, to dissect the dynamics of transcription initiation. In short, if you have a process that requires two consecutive steps and you measure how long that process lasts, in conditions where the duration of one and only one of the two steps differs, it should be possible to infer the duration of the other step using what is called the Lineweaver-Burk Plot, which is shown on the right. Here we did this, and we measured how long RNA production took in conditions where cells had different concentrations of RNA polymerase, because this concentration affects the duration of the first rate-limiting step alone. As shown in the slide, we performed these measurements in various conditions. We started with measurements of the artificial promoter LACARA1, which we call the control condition. Then we changed media. We also used single-point mutant promoters, and we also used different promoters activated by different molecules, etc. As a result of these measurements, we obtained these graphs. They show the mean, the coefficient of variation, the skewness, and the kurtosis of various distributions of time intervals collected. We find that these distributions differ significantly from each other, which is very important because it shows the elasticity of the process. We also find a very strong correlation between the skewness and the kurtosis. This correlation suggests that it may not be possible to regulate them independently from one another. Focusing on the skewness, note that the results on the blue circle show that the same gene, when changing its induction alone, can differ significantly in the value of skewness. Meanwhile, from the results in the red circle with mutant promoters, we find that the genetic sequence of the promoter can also cause skewness to differ. Meanwhile, and finally, from the results in the green circle, we find that different promoters differ in skewness, and that genes differ in skewness when in the stationary and the exponential growth phases, which are very interesting results for many reasons. To assess if one can change one feature of the distribution of intervals within our reproduction events without changing another feature, we plotted each such feature against one another. We find from plot F that only the skewness and the kurtosis cannot be changed independently from one another. This implies that one can change skewness and kurtosis independently from the mean and the variance, which is important from the point of view of how can one regulate this process. Next, we investigated how the skewness and the kurtosis can be regulated so that they differ between conditions. Based on our initial hypothesis, we produced those tau plots, that is the Lineweaver-Burk plots, so as to measure for each condition, on average, how long is it taken by the steps prior and the steps following commitment to open complex formation. For this, as described before, we measured transcription rates in cells with different RNA polymerase concentrations. Having the mean time spent in each step prior to the open complex formation in each condition, which is shown in the green table, we investigated whether these mean times are in some way correlated with the skewness and the kurtosis. From the bottom right graphs, note that the mean times prior to the open complex formation are correlated with skewness and kurtosis when considering, for example, all different mutant promoters, or when considering conditions that differ solely in the regulatory factors. From this, it is possible to conclude that the promoter sequence and transcription regulation allow tuning the skewness and kurtosis of RNA production dynamics. Finally, we verified if protein numbers followed RNA numbers. From flow cytometry measurements of proteins expressed from the target RNA, we find that they do follow the RNA. From this, it is then possible to conclude that the promoter sequence and transcription regulation both allow tuning the skewness and kurtosis of the single-cell distribution of protein numbers. They can also be combined in order to obtain even more complex dynamics. In conclusion, we investigated whether the single-cell distributions of RNA and protein numbers resulting from gene expression in E. coli have asymmetries. From the point of view of the population, we found that they do have significant asymmetries, and also that these asymmetries can be regulated by tuning the kinetics of the rate-limiting steps in transcription. Because they can be regulated, we hypothesized that the decision-making circuits of E. coli may be adaptable to changing conditions by changing the skewness and kurtosis in the protein numbers of the component genes. Similarly, because they are sequence-dependent, we hypothesized that the skewness and kurtosis in protein numbers of these decision circuits, they may be evolvable. For more information, please read the article cited below. Thank you very much.